Hey everyone, it's me, Andrew Rouse. I'm here doing this presentation, doing my part of the presentation, because I am currently playing a baseball game over at McCann. So I'm going to be presenting my portion of our journal club. So my first portion is going to be the background, and obviously so far we know that our paper is mainly on the EGF receptor, which I'm just going to say EGFR instead of EGF receptor the entire time. So at first I want to talk about the importance of EGFR. And its major roles are in proliferation, differentiation, survival, and motility. It does so by activating other pathways in the signaling cascade, such as ERK1 and 2 and AKT, which are going to be talked about later on. EGFR is also important because overexpression of the receptor and mutation of the receptor can cause many of these things like epithelial cancers, glioma, glioblastoma, and carcinoma. There's been a lot of research so far of trying to target this receptor for treatments for the carcinoma aspect of it. This next portion that I'm going to talk about for the background is going to be a little bit of review from class. We've talked about a bunch of these pathways previously. So here I'm going to talk about a little bit of the GRAB2 pathway and how that activates RAS and then activates the ERK pathway. So here we have the EGFR. Upon ligand binding, it dimerizes, creating 2, and then it also phosphorylates the cytoplasmic domain. So this is the extracellular matrix, and it's going to phosphorylate the cytoplasmic domain of the EGFR. GRAB2 is an SH2, so that'll dock on the phosphotyrosine, and that'll bind to the receptor. Upon activation of GRAB2, SOS is a SH3 adapter, and that will bind to the GRAB2, which is going to be a proline-rich region because it has an SH3 adapter. SOS is also a GTP exchange factor, and once that binds with GRAB2, that will switch RAS GDP to RAS GTP, which then activates the RAS cycle. Once RAS is activated, it then changes from, as we learned in class, MAPKKK to, and that gets phosphorylated to MAPKK, and that gets phosphorylated to MAPK, which then is also going to trigger the ERK cycle, which will help out in cell proliferation. Another pathway I want to talk about was the Sybil pathway and the ubiquination of the receptor. So this just shows you a quick slide. This will be the inactive EGFR, and upon ligand binding, this red stick thing that will dimerize the receptor, and then the GRAB2 will then bind, and the Sybil will also bind, is also an SH3 like SOS, so that'll bind to the proline-rich region of GRAB2. Upon binding, that'll add ubiquitin, which are these green dots, to the receptor, and then that receptor will get internalized, and then either they'll be packaged into an early endosome, and then can be degraded into a lysosome, which acts as a negative feedback loop. You're good, Dan, for that first part.